Yeah, hello. Good afternoon, everybody. Yes, I'm going to talk about cells. About cells in 3 dimensions, that's the tomography of cells, and actually about 2 important tools for that process that are electron and X-ray microscopies. So for the next slide, excuse me. Okay, so when I say that we are going to study cells, I really refer to study molecular machines, really DNA replication, a ribosome, how from the DNA we go to the protein at a resolution that is normally a few angstrom, a few nanometers. And we are going to do that with electron microscope, following principles that are very similar to computerized axial tomography. So this is really the normal work we do in the lab, that of course we can go to the cell dimension with tomography, with cryo-electron tomography. What you are seeing here is a centrosome, our first data on that. So if you think about ciliary diseases and all that, this is the molecular basis of many of them. Okay, so we are studying cells and we are using electron microscopy, but we have a problem. Electrons don't go through cells if they are more than, say, 1 micron thick. How many cells do we have 1 micron thick? No, not too many. So if we want to study really normal cells, cells on which we can make a number of interesting questions, pose a number of interesting questions, we have to invent another microscopy. And this other microscopy may be X-ray microscopy. It's a way to get shorter wavelengths, so we really can have shorter wavelengths than normal confocal or optical microscopy, because obviously cellular cell biology is very much studied with optical microscopy, confocal microscopy. The single problem is that you don't have such a high resolution, but X-ray photons are really of a shorter wavelength than the normal you use for vision. So we can get higher resolution, we can go through thicker cells, and we can understand really at the molecular level through labeling which are the proteins we are studying. So this is what we proposed. The problem was that we needed a synchrotron, so we went to a synchrotron, ALBA, in Barcelona. We proposed the creation of a whole beam line. We did it. And now, after the summer, it will become in operation the 3rd X-ray microscope on the world. And, well, the funny thing was that nobody knew how the image formation system was. So which was the concrete and objective relationship between the images and the volumes? Well, we have just published that for some simplified assumption, and we really have quite a field in front of us. So the wish list is very simple. I mean, anybody interested in optics about how to understand the image formation system of a completely new microscopy? Anybody interested in tomography? How do we invert? How do we do a reconstruction? How do we do the from many 2D to 3D volume? Is a very challenging mathematical problem. And of course, which are the biomedical implications or applications? Which cells would be interesting to study at a resolution of between 20 and 50 nanometers? And I finish. So here I am, the Instruct Image Processing Center, located at the Spanish National Center for Biotechnology. Please keep in touch, and you have a complete and whole new field in front of you.